ions like a Z ion with a hydrogen added to it or an extra proton, you can add that as well. So here's just a screenshot of the application. Here we've got the pulled in metadata from both the MS file and the ID file overlaid. In this case, we've got HCD as our dissociation method, high collision dissociation. And so most of the time you can change the ions that you look at if you're interested, but the defaults are B and Y ions when there's high collision dissociation energy. So we see the matched profiles between the calculated and experimental MZ. We also get a little snapshot of the MS1 that this data comes from. So you could see from the parent ion from this deconvoluted, from this non-deconvoluted data where the calculated value is for the entire fragment is where its mass should be and where the instrument selected from. And it's ranked by score. So the best match is going to show up first. And so when you open the app, this is the best match that was in the file and the calculated fragmentation patterns. Every single plot, every single table in this application is easily exportable with this widget here. And yeah, they both have interactive and static modes for all of the plots. So as I mentioned before, that was just one aspect of PSpecteR that you can use dependent on need. There's also lots of other workflow choices in there, including the ability to look at extracted ion chromatograms, which you take your targeted mass and across the entire file, across all of the spectra, you pull the retentions and the intensities, plot those against each other with each of the families indicated here. You have the ability, if you just want to look at the metadata from a file, you don't have any ID data, but you're just interested in looking at what's in your file, you have the ability to plot several different combinations of those variables against each other with labels. We also have the protein coverage, which I mentioned before. So each of these lines is an identified peptide with its scan number and the exact span of the literature sequence of the protein on the x-axis. And if you've included your FASTA database file, which is optional in PSpecteR, you're allowed to see this. And here we've got the name of the protein. And then one of the features that's tightly knit to the top-down tools that we are provided with PSpecteR is the ability to visualize feature maps like from MS Pathfinder. ProMix is one of the algorithms before the actual step where you match each of the identified features to a database. And so in this case, we've got some of these identified features with a log abundance. So PSpecteR lives in 3 different containers. You really only need the Shiny app container if you're not interested in running some of the tools. MS Pathfinder is the top-down tool you could currently use in PSpecteR. So you upload your data, you click run database search tool, MS Pathfinder, that information sent to this container where it's run. And then once that is finished running, it pings you to let you know you're done. There's a backend data folder because these files are large and uploading a 3 GB file through a Shiny environment is slow. So it's just much better to have in part of the Docker Compose structure, this file, that backend folder that you can pull data in and drop data off. And so that's what the entire design of PSpecteR is on Docker Hub, with instructions where you should just be able to copy paste it, and then it shouldn't pull the container for you, and you should be able to run the tool relatively quickly. MSGF Plus is also included for those of you interested in bottom-up proteomics approaches. So as I mentioned, PSpecteR started as a nice visualization tool, which serves as sort of a hub or location. Depending on need, you can do many different quality control plots and visualizations, but it has definitely grown a lot from there. The generation of theoretical fragment ions and matching for spectral annotation, that's been used a lot by Isoforma. In separate projects that are interested in comparing top-down database search tools, I've used the functions on the back end for making those heat maps of the feature identification. And in a separate project as well, I've used the ability to calculate the ion intensities for the MS1s for several different modifications for those interested in more MS1 proteoform quantification techniques, which is not covered in this talk. We are mostly focused on MS2. There's also a vignette included for our package, and feel free to check it out and submit me issue requests to keep me busy. And thank you very much. I'm going to transition this over to Aivett, who's going to get into Isoforma, which has been built based on the functionality in this package. Thank you.
Thank you, David. Hello, everyone. Thank you for joining us today. As Mo and David mentioned, I will focus in this last part talking about Isoforma. And I will start with this example and specifically talking about quantifying isomeric proteoforms. Isomeric proteoforms are basically proteins that have the same precursor mass, it's the same total mass of the proteins. And in this specific case, they have the same modification, the acetyl modification in different combinations, but the positions are different residues. So for example, at the top one, we can see there is a acetyl and the residue number 20, which is in the bottom example, these modifications is at the number at the position, the residue number 8. So as you can see, there are both proteoforms have the same total of 4 acetyl modifications, but the positions in this of these modifications are different, and these combinations might be related to a different protein function, and that's why it's important to study and characterize them. As you can see, the precursor mass is the same, so we cannot distinguish them based on the MS1 level. And one of the approach to study these isomeric proteoforms, which are also called positional isomers, is to use the MS data. As David was explaining, depending on the position of these modifications, we can generate and annotate the fragmentation data, the MS2 data, and we can look for these ions that are specific or unique to these proteoforms, and then quantify the different proteoforms based on those unique ions. There are different tools that have used this similar concept. Particularly last year, our collaborators demonstrated the use of this approach in their publication to characterize histones. The workflow starts with a mass deconvolution, so it means they're using the mass deconvolved data instead of the charge result. And then the next steps in this process was to combine James Pasavento implemented a custom Python script to take this the convoluted spectra and look for the spectra that corresponds to each of the isoforms, to then accumulate this MSMS into a single one and improve the signal to noise. And then other tools were used to validate and assign these different PTMs. And then Excel was used to do the final calculations and normalizations to quantify each of the proteoforms. This approach, as I say, was demonstrated and was used to quantify more than 80 different histones in this work, in this publication. And the limitation was due to the different manual steps that were required at that time. The number of samples that could be processed, it was limited. Then Mowei brought us together and we started this project, the Isoforma project, to further to automate this quantitation algorithm, this process into a single tool. I prepared the proposal last year that was funded and approved by EMSL, and David implemented the tool guided by me and Mowei, and we are working with our collaborators, James Umen, Sarah Romenfacher, and James Pasavento, that will help us to validate the tool and make sure the results are correct. Now I would like to show you a particular example of what's the benefit or the advantage of using the Isoforma. On this slide, we can see the estimated time that we observe it takes for quantifying one isoform in one sample, and it's about 1 hour. And we see there are several tools that are using this in each step, and then the user has to do a copy. There is a copy paste procedure. So we see here that these manual steps require many clicks, and it's a really user intensive manual processing. With Isoforma, what we wanted is to combine this tool into a single software to automate this processing. And we used MS Convert to ProteoWizard to convert the vendor software to an open format, MCML in this case. This takes 3 minutes, and then David implemented Isoforma in R using the functionality from the PSpecteR library to generate the fragment annotation, to generate the theoretical fragments and annotate the experimental spectra. And then with the Isoforma code base, it's combining all the different tools, the processing of all these different tools into a single code source. And as you can see here, instead of 1 hour of processing with the previous demonstrated algorithm, now with the new tool Isoforma, we can take this processing time from 1 hour down to 4 minutes. And another difference is that, particularly for Isoforma, it's that we are not depending on other tools to do mass deconvolution, because in our approach, we are generating theoretical isotopic distributions to match to experimental data directly, and we are using those matches, the scores to filter, as David explained, to filter and down select the fragments, the best quality fragments to do the quantitation. The Isoforma input basically is these 3 items. First, of course, we need the mass spec data. We define a parameters file where the user can set what is the mass, the tolerance, and the correlation score filters, and so on. And finally, we have the list of targets where we specify, the user can specify what are the files that wants to be processed. The protoform group is basically an arbitrary name. Then the retention time window where these proteoforms, the proteoforms, they elute because they have similar modifications. They have same mass. They usually co-elute. They are within the same retention time window. And then the unmodified sequence that will be used to generate these theoretical ions and the fragments. And then they specify the modifications where they are using this notation. You specify, the user specified the name of the modification that is automatically recognized by the backend that is using PSpecteR to generate these theoretical fragments based on the unimode notation. And then basically the user can specify the positions where this modification will be made. And then Isoforma will automatically generate all those combinations. In this specific example, we are using the symbol of that top arrow just to specify that we want the first modification, the acetyl modification, the first residue, it's fixed. So we want all the combinations of the first position in pairs with the next other ones that are up to 2 modifications for this specific example. And then we have the output folder where Isoforma will generate and store the results. Now for the output here, we have an example. Isoforma generates automatically for each of those targets specified, and the runs will generate an annotated MSMS, the MSMS spectrum that is basically all the spectra that were found, and were some accumulated to do the annotation. And then this is also generated in a CSV and MGS format that can be used then to further investigate if the user wants. Visualization of the PTM error that it's helpful for QC. It outputs a CSV with the quantitation matrix, which basically has all the abundance found for the fragments that were accumulated for each of the residues, and the normalizations that were done, and finally the image and the proportions of each of these modifications, the multiple isomeric proteoforms. How is Isoforma available to EMSL users? We are currently working, testing, doing some fine-tuning, refinement of the code, and we are expecting to release it soon, and will be available for the EMSL user community and for the mass spec community in general. And we are preparing a manuscript. Proposals can start requesting using Isoforma already, and EMSL users specifically will have dedicated support and assistance from us for using Isoforma, and also for customizations or additions of features that they might need. And we hope that with this automation, Isoforma will help researchers to characterize and discover new functions of proteins that were not characterized before in large-scale studies. Thank you for your attention, and now we are open to questions.
Okay, thanks, Aivett. All right, well, we do have some questions to get us started, but I would just ask if you have additional questions for any of our speakers, if you'd please put them in the chat, and we'll continue to ask them. First question is for Mowei. Mowei, what is the role of GC content of organism during the digestion process, or even for the top-down approach?
Thanks for that question. I'm trying to make sure I understand it. I think you are referring to more like the gene sequences. So if I understand it right, I think like we are in this process, we're directly looking at proteins, and you may be worrying about like a specific amino acid sequences that may affect the digestion process. I hope that's what you're trying to ask. Because we're using mass spectrometry, and we just detect the masses of those amino acids and in the peptides and also the proteins. So there's relatively a minimum effect of the exact sequence on our analysis. And for the digestion, typically in bottom-up proteomics, we use some kind of a specific enzyme that has a preference on a specific motif or like site. For example, like trypsin, you cleave up the lysine and arginine. So I mean, in that case, you do have issues with specific amino acid sequences, but for the top down, because we don't cleave them, and then the sequence, the exact sequence shouldn't matter that much. I hope I get what you're trying to ask, but if you have additional questions, feel free to send in the chat again.
Okay, very good. Aivett, do you support data analysis workflows other than the specific top-down experiment you mentioned?
Right now, we support this specific experiment that we presented, but as David mentioned it, his work developing the PSpecteR package now will facilitate addressing other kind of workflow, and we are happy to collaborate and help users to get their data, analyze it, and taking advantage of it as much as possible.
Okay. Just a follow-up question. Can you elaborate a little bit about how you would want people to get in touch with you about collaborating?
Well, the first thing is just send us an email, and we are happy to schedule a meeting and discuss. We have several mechanisms for EMSL that we can get funding for their research through the user calls and putting a proposal.
Okay, very good. All right, David, what are the updates being made to the PSpecteR app and package?
Yeah, the updates currently are we're first to support faster searching in general for Isoforma downstream. The original algorithm was just a tiny bit slower, so you can optimize the sort of fragments you pick before you do the matching, and that was added. Plus, with other proposals in the work, there's further development for potentially getting the array of potential fragments to match and then having a more targeted search. So instead of looping through every single combination of PTMs, you look at all of it sort of like as a larger matrix and use that information just to trim down the time. You know, each time you iterate through something, it's slower. So what are all the ways that we can either iterate in parallel or skip the iteration altogether? So that's the sort of updates that have been made. It's been constant tweaks here and there that really do make performance changes and speed long term.
Okay. All right. Here's another question for you, David. To what search algorithms is PSpecteR compatible? And what is your experience in overlap comparing different database search algorithms?
That is an excellent question. I love that one so much. I could say that right now it's built specifically just for our in-house maintained top-down tool MS Pathfinder T and its output, which is MZID. And I have lots of ideas to kind of collect all the different outputs from top-down tools and make it make either a reader that takes all of those and plugs it into the app easier, or use existing tools so that you can make more comparisons in the long run. The good news is tools like TopPick, as it's gained usage and tractability, we could Dockerize that and then just add it to PSpecteR because we already have the design for that in place for the other two search down tools, so top-down search tools. So currently there's only one top-down tool supported, and the comparisons that you would make typically are, if I set this parameter here and this one here, what would it change the output? And how would that change? But I do have extended interest in including TopPick and other tools in PSpecteR.
Okay, great. Mowei, what is the protein size limit of your technology?
So yeah, that's a good question. So for right now, the most commonly used technology, we're usually looking at the relatively smaller proteins like 20 or 30 kilodalton or less. And the histones are kind of the best example. It ionizes really well on the mass spectrometers and it fragments really well. And those are also very interesting targets because they have a lot of PTMs as we're focusing on right now, and it connects with a lot of like epigenetic topics. So that's kind of the best application right now if you're interested in this area of research with top-down proteomics. And also, I want to quickly add to the question to David. For the comparison of different algorithms, yeah, as David was saying, like it's in the progress. Like we're going to like increase the compatibility with other software tools, and some of the like the topic analysis, like we mentioned the topic R. That's another package that's developed by our other colleagues here, and that's more like a focused on topic R. And the overlap between different algorithms is actually not that great. Like you kind of feel like each different algorithm, like they're really good at capturing a specific set of proteoforms by top down. So that's also like important because right now like the top down community is still kind of developing the better tools to kind of automate everything. But I guess that's why it's important to have those visualization tools so we can actually manually look at them and kind of feed the knowledge from manual analysis and improve those algorithms.
All right. Aivett, what is the best approach if I want extra functions to be built?
Yeah, as I mentioned, we plan to release this code open source. Yeah, it's open source, so we will release this code soon. So if you are bioinformaticians or software developer, well, we are happy to collaborate. If you can, we can help you navigate the code. If you want to implement new functions yourself, the other mechanism is to get to submit a proposal to EMSL calls and specify that you need new functions built using Isoforma or PSpecteR, and then we are happy to help with those. And again, please contact us if that's something that you are interested and we can discuss further.
And that's a good reminder that we do have an open call right now. That the letters of intent are due at 5 o'clock tonight Pacific Daylight Time, the EMSL Joint Genome Institute call for ficus research proposals. We have had some technical issues with our website today, so we encourage you to continue to check back throughout the day if you go to the site and have any trouble. David, how was PSpecteR designed to adapt to the evolving needs of the top-down community?
Love that question as well. Well, for example, one of our newer needs is the ability to adapt and add additional tools. So we already have a framework with Docker, and there's some, if you really want to get into the fun of it, there is some like Python code in there too to help submit jobs and return output. So that's a portion there for adding new tools. And then as Aivett mentioned too, the way that PSpecteR is being expanded, one of the things I want to add is a page where you answer the questions of what you need, or if you use the app a lot, you just supply a parameters file. And then for things like, what if you do want to use Isoforma in your workflow? It doesn't currently exist in PSpecteR, but that's something that I'd want to add for sure. And so that's sort of the ways that we take the package and the functionality from the package and build on top of it the GUI layer. So that's really, and on top of that, the Docker layer, so then it becomes OS independent. So there's sort of like a multi-tiered thing I have going on here to try to hit all of the needs at once and try to get everything on the same page as much as reasonable for topics that do fit in PSpecteR. And the overarching umbrella underneath that is the quality control steps within like the ability to quantitate proteoforms. It is a good fit underneath PSpecteR. So ideas are great, send them my way.
Great. All right, that is all the questions that I am seeing right now. We'll give you another minute to drop any in the chat that you might have. And you'll also notice that in the chat, we listed email addresses throughout the webinar today for each of our speakers if you want to reach out and have any additional questions. I'm not seeing any additional right now. So I want to thank our speakers and thank you for joining us today, and also remind you to come back and join us again April 27th for our next EMSL Learn webinar. Take a look in the chat to grab the link so that you can register. And thanks again for joining us. Have a great day.